Good morning, everyone, and thank you for being here today. I'm pleased to introduce our presentation on growth promotion testing of media and cultures. My name is Dr. S.K. Singh, and I hold a PhD in this field. Over the next few slides, we'll explore the key aspects of testing methods to ensure media and cultures promote growth effectively. I look forward to sharing insights and discussing best practices with you all. Let's get started. Let's start by looking at some key definitions related to growth promotion testing. First, GPT, or growth promotion test, is a biological test used to verify that our culture media are performing correctly. Essentially, it tells us whether the media can support the growth of microorganisms as intended. Next, we have positive control organisms. These are specific microorganisms recommended by pharmacopoeias, and they serve as a benchmark to ensure that our media are functioning properly. Then there's the inoculum size, which usually ranges from 10 to 100 colony-forming units, or CFUs, per test organism. This is the amount of microorganisms we introduce to the media during testing. The main objective of GPT is to make sure that each batch of culture media can support the growth of the microorganisms we specify. This confirms that the media are not inhibiting growth and are suitable for use in quality testing. From a regulatory standpoint, performing growth promotion testing is mandatory. Pharmacopoeias such as USB, EP, and IP require this step before conducting sterility tests, microbial limits, or other microbial assessments. In summary, GPT is a crucial part of quality control, ensuring our media are up to standard and reliable for accurate testing. Let's take a look at the key moments when it's important to perform GPT. First, whenever you get a new batch of media, it's a good idea to run GPT to ensure its quality and reliability before use. Similarly, after sterilization, performing GPT helps confirm that the sterilization process hasn't compromised the media. It's also crucial to perform GPT after the expiry date to verify that the media still functions as expected. Another important point is when the media has been stored under non-ideal conditions, such as temperature fluctuations or humidity, which can affect its integrity. Lastly, before conducting critical microbiological assays, performing GPT ensures that the media will support the growth of microorganisms and provide accurate results. In essence, these timing points are all about ensuring the media's quality and reliability at the right moments, which ultimately supports the accuracy and success of your microbiological work. Let's take a closer look at regulatory norms. First, under key pharmacopoeial rules, it's essential to use authorized strains, such as those from ATCC, NCIM, or NCTC. These recognized sources ensure consistency and reliability in microbiological testing. When preparing your inoculum, aim for a concentration of about 10 to 100 CFU, which is enough to detect growth without overwhelming the system. And of course, it's crucial that the inoculum shows growth within the specified incubation period, confirming the test's validity. Moving on to typical reference standards, there are several important guidelines to follow. The USB references, specifically 61 and 62, provide detailed methods for microbial limits testing. The European Pharmacopoeia, EP 2613, also outlines standard procedures. Additionally, the IP microbiology sections serve as valuable benchmarks. Connecting this all to our overall theme, these norms and standards are the backbone of ensuring our microbiological assessments are accurate, consistent, and compliant with global regulations. They help us maintain the quality and safety of our pharmaceutical products. Now, let's take a closer look at typical test organisms. These are the microorganisms we commonly use when conducting laboratory tests to identify and study pathogens. They serve as standard references because they help us understand how different organisms behave and respond to various diagnostic methods. Highlighting these key players is essential because they form the foundation of microbiological testing. By recognizing these organisms, we can better interpret test results and ensure accurate diagnoses. This understanding ties back to our overall goal of effective infection control and appropriate treatment strategies. Remember, knowing the typical test organisms is crucial for anyone working in microbiology or clinical diagnostics. Let's walk through the GPT methodology step by step. First, we prepare our media samples, and it's important to do this in duplicate to ensure accuracy and reproducibility. Next, we standardize the inoculum by adjusting it to approximately 10 to 100 colony-forming units, or CFU, which helps us maintain consistency across tests. Then, we inoculate the prepared media with our standardized sample. After inoculation, we incubate the samples under conditions specified by the Pharmacopoeia, which ensures the process aligns with established standards. Once incubation is complete, we observe the media for any growth. This step is crucial because growth indicates microbial presence or contamination. Finally, we record all our results carefully. This systematic approach allows us to reliably assess microbial contamination and ensures our testing process is both precise and compliant with industry standards. Let's walk through a calculation example to clarify how we determine the right inoculum. Suppose our starting culture has about 1.5 times 10 to the 8th CFU per milliliter. Our target is to inoculate with roughly 50 CFU. First, we dilute the culture 1 to 10,000, which brings our concentration down to about 1.5 times 10 to the 4th CFU per milliliter. Next, we take 0.01 milliliters, or 10 microliters, of this diluted culture. This gives us approximately 150 CFU, because 1.5 times 10 to the 4th CFU per milliliter times 0.01 milliliters equals 150 CFU. Then, we perform a further dilution of 1 to 3, which reduces the concentration to about 50 CFU in 10 microliters. So the key point here is that to get the desired inoculum, we use a 1 to 30,000 dilution and take 10 microliters from that. This ensures we have the right number of bacteria for our experiments. Now, moving on to preparing the inoculum itself, our goal is to achieve about 10 to 100 CFU per tube or plate. The process involves a few important steps. First, grow your culture for 18 to 24 hours to have enough bacteria. Then, adjust the turbidity to about 0.5 McFarland's standard, which corresponds roughly to 1.5 times 10 to the 8th CFU per milliliter. Finally, serially dilute this culture to reach the desired CFU level for your specific test. Remember, getting these dilutions right is crucial for consistent and reliable results. Let's take a closer look at incubation conditions. This is a crucial part of the process because the environment we provide during incubation directly impacts the success rate. Key factors include temperature, humidity, and sometimes even the light exposure. Maintaining the right temperature, typically around 37 degrees Celsius for many biological samples, ensures optimal growth and development. Humidity levels need to be controlled to prevent desiccation or excess moisture, which can lead to contamination or other issues. Light exposure is usually minimized or controlled to simulate natural conditions, depending on the specific requirements. Overall, these incubation conditions are essential for creating a stable environment that supports the growth and health of the cultures or organisms we're working with. Connecting this to our overall theme, proper incubation is fundamental for ensuring reliable and reproducible results in our experiments or production processes. All right, let's talk about the acceptance criteria. When we're evaluating growth, the first thing to look for is clear evidence that growth has occurred. This could be seen as turbidity in the medium or visible colonies forming. It's important that this evidence is unmistakable and consistent with what we expect. Next, we want to check if the growth volume or density matches that of our positive control. If it does, then we can consider that a pass. Essentially, we're confirming that the test conditions support growth similarly to our known positive sample. On the other hand, if there's no growth at all, or if the growth is delayed or inhibited compared to our control, then that results in a fail. Delayed or inhibited growth suggests that something is preventing the microbes from thriving, which could point to issues with the sample or the process. So, in summary, we're looking for clear, consistent growth that aligns with our positive control, and any absence or inhibition of growth indicates a problem. This helps us ensure the reliability and accuracy of our testing process. Let's talk about some common pitfalls and troubleshooting tips to keep in mind. First, if you use too heavy an inoculum, you might get false positives, which can lead to misleading results. On the other hand, if your inoculum is too light, you risk false failures, meaning you might miss detecting something important. It's really about finding that right balance. Another key point is ensuring proper incubation conditions. Temperature, humidity, and timing all play crucial roles in getting accurate results. To help avoid these issues, always use freshly prepared dilutions. This helps maintain consistency and accuracy. And don't forget to include positive growth controls every time. They serve as your benchmark, confirming that your environment and procedures are working correctly. By paying attention to these points, you'll improve reliability and troubleshoot effectively when things don't go as planned. Let's take a look at this scenario involving GPT applied to a new tryptic soy broth, or TSB. In this case, we've inoculated the broth with 6 different organisms, each at a concentration of 50 CFU. The broth was then incubated following the guidelines set by the USB, which ensures standardized testing conditions. The key outcome here is that all the organisms showed growth within the expected time frame, which means the batch has passed the sterility test. This is a crucial point because passing this step indicates that the TSB is effectively supporting microbial growth, which is essential for accurate sterility testing. Once the batch passes, it can be released for further sterility testing of the actual product, confirming that the media itself is reliable. In the example results table, you'd see the specific data confirming growth timelines for each organism, providing a clear, visual confirmation of the batch's validity. This scenario highlights how GPT can streamline and ensure the accuracy of sterility testing, reinforcing the overall theme of advancing quality and efficiency in our processes. Let's start with the key takeaway that GPT is mandatory before conducting any microbiological assays. This step is crucial because it ensures that we're working with a standardized and reliable process from the very beginning. Moving on, it's important to emphasize that we should always use authorized strains and maintain controlled CFU counts. This helps us ensure consistency and accuracy in our results. Next, documenting the method, incubation conditions, and results clearly is vital. Clear documentation not only supports reproducibility, but also helps us stay compliant with regulatory requirements. Finally, implementing regular GPT checks is essential because it maintains test reliability over time and keeps us aligned with regulatory standards. Overall, these points underscore the importance of a structured, well-documented approach to microbiological testing, ensuring both accuracy and compliance in our work. For further support, please don't hesitate to reach out to us. We're here to help you with any questions or additional guidance you might need. Remember, you're not alone in this journey, and we're committed to assisting you every step of the way. Thank you very much for your time and attention today. We truly appreciate your engagement and look forward to supporting your continued success.